In this presentation, we're going to look at the cytoskeleton, a network of proteins that provide support for cells and allow transport for materials within the cell. We'll also look at some components of cells that exist outside the cell membrane. This includes cell walls, the extracellular matrix, and junctions between the cells. The first element of the cytoskeleton we'll look at is microtubules. Microtubules are the thickest components of the cytoskeleton, and they're composed of a protein called tubulin. Actually, the basic building block is a dimer of two proteins, alpha tubulin and beta tubulin. The tubulin assembles into a hollow cord that consists of an equal number of alpha and beta tubulin. Because of its construction with two different proteins, microtubules have polarity. That is, they have a plus end and a minus end. However, in this case, the plus and minus don't refer to charge. Rather, it simply refers to the end that polymerizes and depolymerizes most quickly. Both polymerization and depolymerization happen primarily at the plus end, although it does happen, albeit at a slower rate, at the minus end. Some, but not all, have a structure called a centrosome. The centrosome is the site where many microtubules originate, and it's itself composed of two centrioles that are orthogonal, or at right angles to one another. If the cell has a centrosome, it will be located near the nucleus. The minus end of the microtubules are located here, and the plus end polymerizes outward toward the cell membrane. Microtubules help to maintain the cell's shape primarily by resisting compression. They also provide tracks for transporting materials, and the way this works is really interesting. Biologists discovered a protein called kinesin that serves as a motor that travels along these tracks. Because of its role here, kinesin is an example of a protein called a motor protein. Kinesin has a tail that binds to the transport vesicles, a head that hydrolyzes or uses ATP, and a stalk in between. Kinesin uses energy in the form of ATP every step that it takes as it walks along the microtubule toward the plus end. Since it moves toward the plus end, it's particularly useful for transporting vesicles toward the cell membrane. And apparently it can move at about 375 steps per second. So kinesin can walk along the microtubules toward the plus end, but is there a motor protein that moves in the other direction? It turns out there is, and it's called dynein. Like kinesin, dynein can carry cargo along the microtubule. Dynein also plays a role, along with microtubules, in how cells move using flagella or cilia. Flagella are long, tail-like structures, and when they beat, they push the cell forward parallel to the axis of the flagellum. Cilia, on the other hand, are much shorter, and they generate force perpendicular to their axis. Flagella tend to be present as only a single one or perhaps a few per cell, but cilia often coat large regions of cells. Just a quick note here. Although many bacteria also have flagella that look very similar to eukaryotic flagella, the way that they work and the actions of their motors are quite different, as they have different evolutionary origins. If you look at a cross-section of a flagellum or cilium, you would see what is called the axoneme. The axoneme consists of two central microtubules surrounded by 9 microtubule doublets. These are arranged in a circle and are connected by the motor protein dynein. The dynein uses ATP for energy and walks along the microtubule toward the minus end. However, since the microtubule doublets can't slide past each other, they simply bend. Okay, they bend. And by coordinating this action on one side or another of the axoneme, the cilium or the flagellum bends. Microtubules are also very important for moving chromosomes during cell division, and we'll look at their role in mitosis and meiosis later. The next elements of the cytoskeleton are microfilaments, which are also called actin filaments. They're made up of a single subunit called actin. Microfilaments help to maintain cell shape by resisting pulling or tension. Microfilaments have a motor protein associated with them, and it's called myosin, and like kinesin, walks toward the positive end of the microfilaments. In muscle cells, the arrangement of actin and myosin within the cell allows a lot of force to be generated, and the cell actually shortens as the myosin pulls past the actin. Microfilaments are also involved in cell crawling. They polymerize in groups near the cell membrane, pushing it outward and then allowing it to be filled with cytoplasm. Actin and myosin also help to separate cells right at the end of cell division. It does this by acting kind of like a belt, tightening around the cell until it splits in two. Actin also plays a role in cytoplasmic streaming, in which plant cells move the chloroplasts around near the periphery of the cell. Unlike microtubules and microfilaments, which are made up of very specific subunits, there are a large variety of intermediate filaments. Intermediate filaments don't have polarity like the other two types. Generally, they're used by the cell to maintain its structure and the location of organelles, including the nucleus. Intermediate filaments are much less dynamic than microtubules and microfilaments. They tend to remain static for long periods, including after cell death. In some cases, like keratin, it can be excreted by the cell to form hair and fingernails. Outside the plasma membrane of animal cells is the extracellular matrix. It consists mainly of glycoproteins, which are proteins with oligosaccharides attached. There are a number of different fibers, but they generally serve to protect the cell, give it structure, and in some cases aid in the communication of outside the cell to inside the cell. In addition to maintaining a fibrous network outside the cell, cells form connections with each other that allow them to communicate. In plants, these connections are called plasmodesmata. They form between cells where there are pits or holes in the cell walls of both adjacent cells. The cytoplasm extends into and all the way through these plasmodesmata so that the two cells are essentially linked up with a single continuous cytoplasm. Because the cytoplasm is continuous between them, the cells can exchange water, ions, small molecules, and in some cases, larger molecules such as RNA. In animals, gap junctions, shown at the bottom of this figure, serve essentially the same purpose as plasmodesmata in plants. They form from a protein called kinesin that form small pores. When the connexins from two adjacent cells align, they can pass ions and other small molecules between them as a way of communicating. Desmosomes are sort of like spot welds that hold cells very, very tightly together. Glycoproteins that extend through each of the cell membranes interact and hold each other tightly in the space between the cells. On the inside of the cell, they're connected to the keratin intermediate filaments, which helps in giving the cell structure in relation to neighboring cells. Lastly, some animal cells have tight junctions, which is sort of like sewing the two cells together. Tight junctions prevent any fluids from escaping the body via the space between the cells. These are particularly important in your epithelial cells and make our skin waterproof and capable of retaining moisture.